This is a video overview of our paper titled Protein Structural Superfamily Classification using Handcrafted and Language Model Features, a performance versus interpretability trade-off, accepted in TMLR August 2026. Protein structures are often segmented into domains, which are subunits of a protein structure that fold independently of the rest of the structure. CATH is a public database of protein domains. It categorizes protein domains based on similarity of their structures at four hierarchical classification levels. These are class, architecture, topology, and homologous superfamily. Based on this hierarchy, it assigns a corresponding four-level code to each domain. Some are illustrated in this image. More than 500,000 domain structures are classified into around 6,600 homologous superfamilies in CATH. In this work, we ask, can we predict the Cath homologous superfamily of a protein domain in an interpretable manner? Can we gain insights into the characteristic features that distinguish a given superfamily from others? To answer these questions, we train 45 one versus all linear classifiers for 45 different Cath homologous superfamilies. These 45 were selected based on the availability of sufficient samples. Each classifier is trained to predict if a given domain belongs to a given superfamily or not. To train the classifiers, we compute different types of feature vectors encoding varying levels of information from the domain's sequence and structure. We engineer 9 types of feature vectors. Some of these are handcrafted from the sequence, some of these are handcrafted from the structure, and some are extracted from the sequence using protein language models. While the handcrafted features are interpretable, the protein language model-based features are not interpretable. While we train one versus all classifiers for only 45 superfamilies, the all in one versus all includes more than 6,000 other superfamilies. Let's look at the predictive performance of the different feature types in the one versus all classification task. The figure shows for each feature type the average AM score on the test data of the 45 one versus all classifiers. We see that the protein language model-based features have higher predictive performance than the handcrafted features. However, the proposed handcrafted structure-based CSIC features offer a balance of interpretability and predictive performance. But what does the interpretability of handcrafted CSIC features offer? To answer this, we use the marginal contribution feature importance or MCI framework to score the individual features of the CSIC feature vector for a given one versus all superfamily classification. We look at the MCI scores of the CSIC features for a given one versus all task, particularly for the superfamily 2, 130, 10, 10. The y-axis in the figure shows MCI scores, while the sequence separation intervals on the x-axis are the CSIC features. These correspond to the separation in the sequence between residues that form contacts in the folded protein structure. We find that the interval 4 to 18 has the highest MCI score. Let's look at this contact interval in the contact maps of the 2, 130, 10, 10 superfamily domain structures. The contacts with sequence separation in the interval 4 to 18 are highlighted in the contact maps. We see that the domain structures belonging to superfamily 2, 130, 10, 10 are rich in contacts with 4 to 18 sequence separation. These are characteristic contacts of antiparallel beta strands, which are present in beta propeller structures belonging to this superfamily. In conclusion, the protein language model-based features have high predictive performance in the one versus all cat superfamily classification task. But the specific characteristic features are not revealed due to the non-interpretable nature of the feature vectors. On the other hand, the handcrafted structure-based CSIC features offer a balance of performance and interpretability, as the CSIC features are rich in information about the sequence separation at which the contacts are formed in the structure. Finally, the MCI scores of interpretable features, such as CSIC, in the one versus all classification setup, offer a way to identify characteristic features that distinguishes one superfamily from all others. Thus, we illustrate a performance versus interpretability trade-off between handcrafted and language model features in the cat superfamily classification task. Thank you for your attention.